Hello and welcome to today's webinar on non-antibiotic control of fire blight by Ken Johnson, Rachel Elkins, and Tim Smith. This is Alice Formiga and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. You can find all of our published articles, videos, and our many upcoming and recorded webinars on organic farming and research topics on our website at extension.org/organic_production. Before we begin, I just wanted to give you a quick rundown on how today's webinar will work. The presentation will last about 45 minutes, and after that we'll have an additional 30 minutes for your questions. We'll be reading the questions out loud after the presentation is over, and we will answer as many as we have time for. Today we're very glad to welcome back Ken Johnson, Rachel Elkins, and Tim Smith, who have presented several other webinars on this topic which are available in our archive. This group has been involved with fire blight management for over 20 years. Ken Johnson is a professor of plant pathology at Oregon State University in Corvallis, Oregon. Rachel Elkins is a University of California Cooperative Extension Farm Advisor and is located in Lakeport in Lake County, California. And Tim Smith is a Washington State University Cooperative Extension Area Agent and is located in Wenatchee, Washington. So with that, I'm going to hand over the screen control to our first speaker, Ken Johnson.
Thanks, Alice. So I think most of you listening have probably heard us talk about
Muted.
before, and what we've made today is a fairly short presentation so that we can answer a lot of questions. This is the first year that antibiotics will not be allowed in certified organic production, so we think that there's probably some questions out there. We have some fairly detailed recommendations, and they're fairly lengthy and fairly detailed, and I know that for everybody's situation that recommendation probably isn't going to fit. So we're going to sort of talk about that, and then you can ask questions about how it fits for your particular situation at the end of the webinar. So Rachel and Tim will come in here in a second, and I put our pictures on there just so you know what we look like. I think most people know what we look like. And at the end on the handout, I put our email contact, so if anybody has any questions afterwards, they can get a hold of us that way. So, of course, I think everybody knows this by now. Streptomycin and oxytetracycline are now no longer allowed in certified organic production. So this is the first season with non-antibiotic methods that must be used in certified organic. And there's a lot of things we could talk about. We could talk about cultivar resistance. We could talk about cultural management and things like that. And all of us have a lot of knowledge about that. But most of our emphasis has been on protection in susceptible orchards, given that the spray programs have to change this year. So our research has really been focusing on spray programs in susceptible cultivars because we have so many organic blocks that are out there that are susceptible and need to be protected. So that's our emphasis, and that's what most of our discussion is today, though we can field questions on any topic in fire blight at the end. Okay, so this is a slide we've shown many times. Everybody's probably seen it about the fire blight disease cycle, and of course everybody knows that the disease overwinters in these cankers left from last year, and this is a pretty obvious one, and hopefully most of these get pruned out in the winter, but there are less obvious ones that don't get pruned out, and that's how the disease jump starts. It's the next season, and those old cankers for the most part are a dead end for the pathogen to start disease in that place again next year or in the coming season. So the bacteria has to jump out of these old cankers into the flowers, and so we've got this little jump shown here, and it gets in the flowers, and then once it's in the flowers it can grow and multiply very rapidly, and this is a very temperature dependent process. So this is what we call the floral epiphytic phase, and this goes around and around during bloom, facilitated by temperature in increasing how the time or decreasing the time it takes for bacteria to double, divide and split, and also it facilitates the bee activity in the flower. So as temperature goes up, the bees fly faster, and then this goes around and around. So when you get into really warm conditions, the bacteria are doubling quickly and the bees are flying faster, and then this builds up the bacteria in the flowers. And then toward the later stages of bloom, you get enough bacteria in enough flowers that you have a possible infection event that occurs. and a dew or a light rain then can move the bacteria down from the stigmas where it's been growing, and the stigmas is the same surface that the pollen grain has landed on, or pollen grain lands on and germinates and then fertilizes the flower. So the stigma is at the top of the style, and then so this light rain or dew slides the bacterium down the style into the floral cup, and that's where infection occurs in the little openings in the floral cup, and then gets you into infection phase. Then you get lots of ooze on infected tissues, and it gets you into secondary phases during the summer where ooze spreads around by insects and by rain and wind and things like that, and gets you into the secondary phases. So staying clean is sort of the goal of fire blight management, and so a lot of emphasis is placed on the bloom period, because the bloom period is your key step in getting an orchard through that period clean, and then you'll have a lot less problems with it as the summer goes on. All right, so we're going to talk about materials here today. So fixed coppers, Rachel will talk about that, and it's a study that we've done in California with fixed coppers and looking what effect it has on sanitation of the fire blight pathogen. Talk a little bit about lime sulfur. In the West, we use lime sulfur for thinning flowers. So lime sulfur is a very caustic material, and it's used to burn out flowers, and then which thins flowers and reduces the number of fruit you have, but then you end up with bigger fruit. So we use a lot of that, particularly in early bloom in the West. Talk about biologicals, and what I mean by biologicals are the actual materials that grow on the flowers, and if the biologicals get growing on the flowers like the pathogen, then they grow and spread, and they slow the pathogen's growth down by competition. And then we'll talk about materials that we think are appropriate in the place of antibiotics. So the timing of antibiotics would be in this sort of late bloom timing. So bacillus-based biorationals, Serenade Opti is the material that we've worked with the most, or the soluble coppers like Cueva or Previsto, for which the registration is still pending, would be another material that we think is very effective at this stage. And then a little bit about summer control, but generally for organic producers in the future, it's going to be some type of copper that's primarily used in the summer. So we've made this recommendation, and this is very detailed, and I apologize for the small type, but it's sort of the thing that we put out there, and people can look at it and have it in their hand and see what fits for their programs. So the first thing is just what we said, fixed copper sanitation, which we'll talk about in a minute. If fire blight was in the orchard last year, and generally the rates are in this 5 to 6 pounds of a copper hydroxide type of material. Badge X2 is one we've worked with, but there's other materials out there that do the same thing. We don't really feel that the brand of copper material is really that important. And Rachel will talk about this in a minute. And this practice only works if you had blight in the orchard last year. It has no effect on orchards that didn't have blight in them last year. So that's the first step. Second step for us in the West is we do use a lot of this lime sulfur, and the sort of the typical protocol is king bloom open in apples, and then a second one about 3 days later, and there may be some others. There's a lot of variation in these programs. But these programs we found are pretty effective on the fire blight pathogen, and while you're putting these lime sulfurs on, they're suppressing the pathogen's growth in the flowers, and for that reason we like them. The thing about lime sulfur is that they are very hard on biologicals. So if, for example, if we're using Blossom Protect and lime sulfur goes on the top of it, we're doing a really good job on wiping out our Blossom Protect yeast in the flower. So we would like to see that the biologicals get reapplied if lime sulfur goes on after the biological. And then so more getting into the program here, how to use a biological. And so let's say we put on 2 lime sulfurs in apples, or we're just starting in pears. We would put on Blossom Protect first, and the sequence is very important here. Blossom Protect, and then that would be, say, 1 full, or what we call half applications, which would be alternate row spraying, so that the whole orchard gets covered once, 2 half applications. Or if blight was in the orchard last year, 2 full applications if blight was in there. And then in apple, this first Blossom Protect treatment would occur immediately after the second lime sulfur treatment. And a little bit of a problem we've had with some very russet-sensitive pears, which are angios and camise and some things like that, and in a little bit wetter climates, there is a bit of russet risk with Blossom Protect, so we'll talk about that. And if you're growing for fresh pack pears for a graded fresh market and russet is a concern to you, there's a bit of risk with Blossom Protect, not a lot, but a bit, and so we're kind of just making people aware of this so that they know this. And so the alternatives would be Bloom Time Biological is an alternative, and it's one of the bacterial biocontrol agents, and it's generally considered a fruit-safe material. Then finally, getting into now full bloom to petal fall, and this would be the traditional timings of antibiotics. Serenade Optimum, or Serenade Opti, I think it has a name this year, is the most fruit-safe material. It works like oxytetracycline works. It's not quite as effective as oxytetracycline, so the interval becomes important, and that it be applied a little bit more frequent than an antibiotic would be applied. And you get improved control if you mix Serenade Opti with Quava, and the rate can be 2 to 3 quarts, which is not probably the most effective rate by Quava by itself, but mixed with Opti, we feel like we're getting some response out of Quava. And the problem with Quava is that it's a copper, and again, coppers on fruit or on flowers in late bloom can lead to some russeting problems, and we've seen a little bit of that in our trials, which we'll talk about. So that's the concern of working with coppers in late bloom. Okay, I'm going to turn this over to Rachel now, and Rachel's going to talk about this delayed dormant study that we did in California for several years, for 4 seasons, and I'm going to turn it over to her and let her talk, and she'll just tell me when to click the slides.
Okay, I hope you can hear me. I'm on my telephone, so I may not come out as loud as those who are using their headsets with their mic and speakers, so kind of just signal that or something if you can't hear me well. So anyway, the work that I'm going to talk about, a lot of you actually have probably already heard about this. This is the application, going back to that previous slide that Ken first showed, where he had the epidemiology of the disease. The first step was the disease surviving in the overwintering cankers, and then jumping from the cankers being taken by insects over to flowers or by rainfall, going from flower to flower. So basically, the idea here is that applying a delayed dormant copper just prior to green tip, and the reason we chose this timing was just that it was a little bit safer. However, if you're feeling brave and you are not so concerned about russeting, for example, if you're straight processing or just russeting is not an issue for you for some reason, then the closer you move this treatment to flowering, the more longer the benefit you're going to get from it. But for safety's sake, we chose the delayed dormant treatment, which is just before the green tip stage. So it's the application of a high rate of fixed copper to the orchard. It's a full coverage spray. And we used generally what it is, it's a Pseudomonas application rate on the label of most of these materials. So when we first started this, we were using a product called Coside 3000, which was being used in California, and we just switched over to another product called Badge X2 because it just on the cost side was less expensive, but they both are in the 28 to 30% metallic copper equivalent fixed coppers. So one of the things about this is that you're not going to be using those new soluble coppers in this treatment window. This is for the higher metallic copper equivalent coppers, not for your very, very low soluble coppers. For this, you need this material, this treatment to last for a while in the orchard to suppress the pathogen, the inoculum, and so you need some longevity. You need some more copper, a higher amount of copper ions out there. So this material is applied. We applied it along with the normal delayed dormant oil. So our treatments were essentially the copper tank mixed with the oil and the oil alone, but the oil plays no role in the fire blight control. It was strictly a convenience to put the copper in the tank with the oil that was already going on the field. So this is the rate that we use. Ken said 5 or 6 pounds. It just depends on the material that you're using. Just make sure that pears or apples is on the label. That's what you need to make sure of.
And it's OMRI listed.
And it's OMRI listed. Yes, it's OMRI listed, and that will be a restrict you to which material you're going to use for this purpose. Alice, so do you want to? So what we did, and I guess I'll continue here. So that's the go back, Alice, again to the left. Okay, so on the left side of the screen, I explained the treatment. On the right side of the screen, what we did to show, so the idea here is that we are going to, it's basically a sanitation treatment. What you're doing here is in orchards that you might call dirty, i.e. there are holdover cankers from the previous season, where you suspect that you know that you didn't do your pruning job did not take all the holdover cankers out. That's the orchard we're targeting here. So what we did was when bloom came, we actually walked up and down the fields and collected samples of blossoms throughout the bloom season, throughout the fire blight season, and we sent those up to Ken's lab, and Ken ran essentially a PCR test on them called LAMP, and he was able to determine whether there was fire blight pathogen or inoculum in the flowers through the season that we sent him, and when that and look at the rate of increase in how those flowers were infected with the pathogen through the season, so we could tell when during the season we were getting fire blight inoculum in the field, and importantly, if there was a difference where we treated with the copper versus where we treated with the oil alone or did not treat with the copper. Okay, go ahead. So Ken in the lab, he used this test called LAMP, and we sent him the bag of flowers, and he ran the assay, and he could detect the DNA of the pathogen. Go ahead. We did this several times during the season. Don't forget, we treated in just before the flowers came out, or delayed dormant, I should say, which was about a month before bloom. And then when the flowers came out, we started sampling, and our first sample was in the mid-bloom. About 50% of the flowers had opened, and then again at full bloom, and then again at petal fall. In some cases, if we had an extended petal fall, we did a second sample, and the petal fall would also equal when the rat tails were coming out. And what we found out was that you can see in this these 4 boxes, the little diamonds are where we put the copper in with the oil in the delayed dormant, and the little squares are where we only put the oil, so no copper. And you can see that the little diamond lines are lower, have a lower amount of positive samples of fire blight DNA in them than where there was no copper treated. So we could show over a period of 4 years that the copper was actually suppressing or reducing the amount of fire blight pathogen in the flowers. Not only did it reduce the overall amount in the flowers during the season, but it delayed when the pathogen was building up in the flowers. So you were actually delaying you were delaying the cycle of pathogen in the field into a shorter window of time, which would mean your spray season was shorter. And this was very apparent in the first year, 2010. In 2011, you saw the same pattern, about 20 to 30% less inoculum, and you have a delay, you can see, of almost an entire period of bloom, from mid-bloom to full bloom, of when you were getting inoculum buildup. Now, in 2012, which was a cold, wet year, there was very little inoculum for a long time, and there was no difference in treatments. In the year 2012, basically what happened was, don't forget this is a prophylactic treatment, and so because it was such a long, cold, wet year, you had two things happen. First of all, you had a very long period between when you put the copper on and when you finally bloomed. And second of all, with all of the extra moisture, your copper ions were basically decreased by the effectiveness of the treatment was decreased by the time it bloomed because of all the wetness that you had dissolving, taking the ions out into solution, and then they wearing away, the copper wearing away by the time bloom came. So we repeated this. We had been successful in 2010 and 2011. We repeated it in 2013 after the 2012 year, and again we got the same results as 2010 and 2011, where the copper both delayed the buildup of inoculum in the flowers and also reduced the overall amount. And the other thing that you can see here is that that when your inoculum builds up in the flowers really doesn't happen so much until the warm part of the year. Really, you start to see it in the full bloom, but mainly in the petal fall. You really see the increase in inoculum during the latter part of the bloom season, not early. Early in the season, there really isn't much inoculum. So what this shows is that your spray program is most important during the latter half of bloom and into rat tail, rather than in mid-bloom and to full bloom. That's where you have the least amount of inoculum actually present in the field. Every year will be different. This year we're blooming very, very early. It was warm. Sprays are starting early. But as far as and so we don't know what the next few weeks will hold, but the pattern should be the same, but it could occur earlier in the season this year. Anything else anybody wants to add about this 4 boxes?
I was just saying that these are commercial orchards, and there's about 14 in each box there, and there's antibiotic programs going on, so the increase in the pathogen is occurring, in spite of antibiotic. A lot of these were, this was done, in some conventional and some organics, but antibiotics were being used. So antibiotics don't necessarily, they're probably slowing, but they don't necessarily stop this epiphytic phase that occurs, and these are commercial orchards. So the other thing is that I don't think I put the slide in, is that we did a lot of fruit finish evaluations on this as well, and we didn't show any effect on the finish of the fruit in terms of russeting by the copper treatment put on at the delayed dormant timing.
Right, that is correct. Steve Lindau at UC Berkeley did all the fruit finish evaluations, and we never found any finish problems using this strategy. And yes, we did multiple. These were grower applied with commercial sprayers. Applied, the orchards were in Marysville, California, Yuba City area, and then Lake County and Lake County, California is where these were applied. So quite a few different orchards each year. Okay, I think that's about it.
So that was just a summary there. So in all these samples we took, and there was a lot of them, there's hundreds and hundreds, that what Rachel was saying, we had less than a 5% probability of finding the pathogen in that early mid-bloom sample. But by the time we got to petal fall, it was at 50% of all the samples had the fire blight pathogen in them, and the copper and oil had averaged overall was half as many positives as the oil alone. So just kind of just sort of a summary of this. Next slide, Alice. Okay, so that's the early part of the season, and now going back over this, now we're into the flowering period, and what we call integrated control. And this has been this has been a concept for us that's developed, over the years, and just spraying a lot of things at flowers. And it's a long story. I could probably do a whole talk just on integrated control. But the idea is that these biologicals grow on the stigmas, and when they grow there, they compete with the pathogen. So when the bee comes into the flower and rubs its body against these stigmas, the pathogen grows there. It's like a little petri dish in nature, I call it, and you can easily grow 1 million pathogen cells on those stigmas in a very short period of time, and that's essential for the pathogen to do that. So if you can get a biological agent on those stigmas in early bloom, you can really slow down this process of the pathogen growing on those stigmas. And that's really important because the pathogen needs a certain number of cells, and we think it's somewhere between 100,000 and 1 million cells on a flower in order to have enough to slide down the style and get down here and actually cause an infection. So we've found over the years that biologicals, and this would be the kind of biologicals that grow on the flowers, like Blossom Protect or Bloom Time Biological or Blight Band 8506, do their best work early in bloom and growing on the stigmas. And then when we get to that later part of bloom, between full bloom and petal fall, we want to fill this cup with some material that is antagonistic or toxic to the fire blight pathogen, and we find that chemical materials of some sort at this point do their best job. And this could be an antibiotic, it could be Serenade Opti fits this role, and we tend to call Serenade Opti, though it's considered a biological product, we call those biorationals because they're more like a BT type product, where the chemicals that are made in the fermentation of the bacillus and Serenade actually are what are doing the work. So there's these antimicrobial compounds, molecules made by the biological organism in the fermentation that grow in the fermentation. And when you spray Serenade, you're filling this little cup with those molecules, and that inhibits the fire blight infection and sprays and prevents infection. And that's the same with copper as well. And the other thing about Blossom Protect, the yeast, it also will grow here, whereas biologicals, the bacterial biologicals like Bloom Time Biological, which is a Pentoia glomerans, doesn't like this area of the flower, the floral cup, because it's very sugary, and bacteria don't respond well in a real sugary environment, whereas yeast do a fairly good job. So that's integrated control, and I put some examples here and why we think this. The original concept was Bloom Time Biological, then Oxytet, but we're in the no antibiotics era now, so analogous concept would be Bloom Time Biological, then Serenade Opti, or in our detailed recommendations, it would be lime sulfur, Bloom Time or Blossom Protect, and then Serenade Opti or Cueva Copper, and these have been giving us excellent control, as we'll see. All right, next slide, Alice. All right, I'm going to turn this over to Tim, and Tim has worked with all these materials, and I've got about 4 slides here of Tim's data, and Tim is the trial guy. He's got so many trials and so much data. I'm just going to turn it over to him for the next 4 slides.
Yes, thank you, Ken. It's very difficult to do one test of one product and come up with a good idea of exactly how effective it is. So what I've done in the next few slides is I've looked at some years of work and then summarized it so that you can see the trend with the product rather than just 1 year's work or just a limited work. This is a summary slide that I usually put at the end, but I like to show this slide to show people that they're not left in the lurch without antibiotics. What the blue bar indicates is the range of percent control that I've had in the trials that I've run. And that number by that blue bar is the number of trials that are in that range. So to take the top blue bar, it says 23. Well, there's 23 tests of Blossom Protect in either apples or pears that I've included in that bar. And the red hatch is the average of those results. And so you can see that oxytetracycline, the fourth bar down there, is the standard that we've used for years as an effective product that that will control fire blight in nature very well. But if you look at the summary slide, you'll see that Blossom Protect, the yeast, and Provisto, a copper product, and Coeva, another copper product, that's both of those are low copper products, are at least equally effective with oxytetracycline and Coumadin, a new antibiotic that's out. And that Serenade in my trials has been good, but not as good as those 5 products above it. And then other coppers have been less effective. So I think this indicates strongly that we have some products that are at least equal, and in my trials, very often better than Oxytet. Now, streptomycin, I haven't included that in here because we trying to cheat and use a streptomycin susceptible strain of bacteria in the test when we inoculate, and it may not be this indicative of what results we would get out in the field where some of the bacteria are resistant to streptomycin. So I've left that off this. Now this is a summary of most of the good results that I've had on Blossom Protect, the Oreobasidium pullulans yeast. And this was the product that surprised me the most over the years. Those red bars are examples of oxytetracycline results through the years. And I think that anything that controls fire blight at this percentage of about 70% or better is excellent. And you'll notice that Oxytet, a product that we've always relied on, and it is very effective if you time it right, is clustered there around 70 some odd percent control. Now what percent control means is it's a comparison of the untreated check that's inoculated to the treatment. And that means I can compare results from one year to another because the untreated check varies from 45 to 60% infection. So I switch around and call it percent control so I can compare one year to another. But you'll notice that some of the best results that I've ever had in the trials are with the full rate of Blossom Protect, 2 applications or more, and with the Buffer Protect buffer in there. I've circled the bottom area down at the bottom of those bars, the ones that are dropping below 70% control, because most of those I didn't include the buffer. You see no buffer, alternate buffer. I've tried using mineral buffers that lower the pH, and they just generally didn't work. So when I followed directions and put on 2 or 3 or 4 applications of Blossom Protect, even in an inoculated trial where we were jamming that at least the minimum amount of bacteria necessary to infect 50% of the blossom clusters right into the flower, no natural infection, just a brutal exposure of the flower to the bacteria. We were getting almost complete protection from this Blossom Protect. And that came as a surprise to me, and I'm still amazed every year when we get the consistent results of how effective this product is. This is a summary of the coppers that have been in the trial. Not all of them, because there's so many copper. There's a lot of different brands of copper. There's a lot of different compounds of copper. But we've done a lot of work on the copper forms that are lower in copper, the 2 or 3% coppers that are formulated in a manner that seem to make them safer to apply in regards to russet, and also they seem to be very effective. So if you look at that range, a lot of the products that are not circled at the bottom, the ones circled at the bottom are the low rates of Coside and the low rates of Provisto, and those are just inadequate rates. But when we get the rates up to adequate, and not that excessive really, the 3 quarts of Provisto or 3 quarts of Cueva, we get very good results. And this is copper compound sprayed on the flower the day before and the day after inoculation. So the coppers have generally performed very well. We're mostly concerned about how you can put copper on safely without russeting your fruit. But as far as the efficacy, it seems to be there. And this is another category of product, the Bacillus subtilis products, that also have done fairly well. They're in general, they're not quite as good, they're not as impressive as the coppers and the Blossom Protect, but they, for the fact that they're very safe on fruit finish, I think that these are going to be very useful products. And you can see most of the treatments over the years have hit in that 70% or better. And it's been my impression that these products have been improving over the years. So I've tended to get better results over the past years than I did early on, years ago when they first came out. They've strengthened many of these products. So you can see the average Oxytet treatment over the years is about 80% control, and we tend to hover in the 70 to 80% control range with these serenades and double nickel. There's a product called Companion up here that is a home garden product, and I only tested it once, and it seemed to work pretty well, but it's very expensive. It's formulated for the home gardener. So that's a summary of the work that I've been doing. I've tested a lot of other products. Some of them are very good, but just for the products that are registered and available right now for use this year, that's the summary that I had to give.
Okay, let me just jump in here and say a couple things about lime sulfur for people that are using it. And lime sulfur does a few things out there. I think you could click advance it one, Alice, please. I think so lime sulfur does a couple things out there. It thins flowers, so the primary effect of lime sulfur, and we're using lime sulfur here mixed with fish oil, and they're both at 2%. Though there's many people that use lime sulfur straight and at higher percentages. So there's a lot of different ways of thinning, and those are kind of an art to it. And it's it gets complicated. But here we're using 2% lime sulfur and 2% fish oil as the treatment. And so the first effect of lime sulfur is that it thins flowers. And so in this chart right here, you can see where we put the kind of olive green bar here in the center of these charts of these trials is that lime sulfur treatment, and we didn't put the pathogen on until after the second lime sulfur treatment. And lime sulfur is a very short residual. It kind of probably within a day has lost most of its punch out there. So you see this effect in control of fire blight, and in this case the effect is primarily, I think, just that there's fewer flowers left that are actually infectable after the lime sulfur treatment because you've burned out the stigmas in these flowers. The second effect, which I'll show you in a slide, following slide, is that lime sulfur is actually toxic to biologicals, including the pathogens. And so here's the population size of the yeast, Blossom Protect yeast, after we put on different rates of lime sulfur on the flowers. So you can see we have untreated control, that's UTC. lime sulfur and fish oil is each at 2%, and then just straight shots of lime sulfur, 3%, 6%, 9%, and then a non-organic ammonium thiosulfate treatment, which is also used for this. But you can see that in the process of putting this material on top of a biological agent, such as Blossom Protect or Bloom Time Biological, or the fire blight pathogen, that we're doing a pretty good job of smashing on them pretty good. That's the best way to describe that. And so we're taking populations down pretty far. So when we talk about people putting on thinning treatments early in bloom and worried about their fire blight control, if they're using lime sulfur, we feel like they're probably doing a pretty good job at fire blight control early in bloom. If they want to use biologicals and they're putting full lime sulfur sprays on top of those biologicals, well, they're also doing a pretty good job of cleaning out their biologicals. So we're recommending that biologicals be applied after the second lime sulfur treatment at least, and then reapplied if there's a need to put lime sulfur back on top of these biologicals. Next slide, Alice. So I would say a little bit more about the yeast. I mean, Tim showed you a really nice summary of the yeast, and it's a really interesting material, and as Tim said, probably the most surprising material that we've ever worked with. And I think both Tim and I, when we were first approached about working with this stuff, said, okay, we'll give it a try, but don't like to bang up trees, and it's expensive to bang up trees. And then we put it on and it worked, and we were somewhat surprised. And so we've done sort of some coordinated trials amongst the group here on the left-hand side there. That's Todd Temple. He's my technician. And you see Rachel and Tim there. And we just did a simple trial in Corvallis, Lakeport, and Wenatchee, where we put on Blossom Protect and Buffer A once at 20% bloom, once at 80% bloom, or twice at 80% and at 20 and 80% bloom. And then we just measured flower, looked at the flowers between full bloom and petal fall. This is the period, when we think most of the infection is happening between full bloom and petal fall. And we sample these flowers, and you can see that the yeast is going on to open flowers at 20% bloom, but it grows on that flower, and a bee comes along and spreads it to the next flower, and that by the time you get into this period between full bloom and petal fall, essentially every flower has picked up a population of the yeast. You can see that the green bars are pears, the red bars are apples, and on the left-hand side that's Corvallis, and on the right-hand side that's Rachel's pears in California, and Tim's apples in Wenatchee. And you can see just how the one treatment of Blossom Protect is basically giving us a very high level of colonization of all the flowers in the orchard. So that's where we're basing our recommendation. If fire blight pressure wasn't real high in the orchard last year, we think one treatment of Blossom Protect is probably sufficient. But if fire blight was in there last year, we think then a second application is probably warranted. So you can sort of see where we get our recommendation from. Next slide. And just so I put a few slides here where we're just putting integrated control together here. Again, this is a similar slide to Tim's, where we have different trials, and I've got one of Tim's trials there in apples, and an apple trial of mine, and a couple of my pear trials. But basically, these are two treatment trials, except for the strep is only put on once. But two treatment trials of a Bloom Time Biological and Oxytet. You can see it's not quite as effective a treatment. But Blossom Protect and Provisto Copper, and it's just really an outstanding treatment. It's as good as streptomycin in these trials. So the numbers on the bottoms of these little boxes are the number of strikes that we're seeing on the trees. So 30 strikes in the Bartlett pear 2013, and in the second Bartlett pear we were up to around 82 strikes. So you can see that these were pretty heavily infected trees, and the degree of control that we're getting from a Blossom Protect and then a soluble copper is really outstanding. Next slide. Oh, wait, one other thing I want to say here. I got a little bit of animation here. So when we use Blossom Protect, and so Tim could maybe comment about this at the end, Tim's developed the Cougar Blight model, which was used to time antibiotic applications in fire blight control. And I just want to make a point that I think that if you want to use Blossom Protect, you're going to have to make that as a strategic decision, and it's not really model dependent. In that, okay, I'm going to base my fire blight control program on Blossom Protect. I'm going to go out there at mid bloom, 50% flowers open, and put this material on. And it's going to really be model independent. You know, it isn't a decision that I can wait and say, oh, the model says today the risk is higher, the risk is low, and get good control with this material. We think it needs to be out there for early in bloom, and that you just have to make the decision to make that treatment or not. But once we get into that, after that treatment's put on, if we talk about the use of Provisto, well then Provisto or Serenade or these other materials are the same as antibiotics in our minds, in that you can put them on and be weather dependent and model dependent. So looking at the model, what does the model say the fire blight risk is today, and then time materials of Provisto or Quava or Serenade based on that. All right, next slide.
If I could,
jump in.
Make a quick statement about that. When you're watching the forecast portion of the model, it's often easy to see the danger period coming in 3 or 4 days. And it's the forecast part of the model that's most important to organic growers, because if you see that it's going to be dangerous in the next 3 or 4 days, and that the temperatures are building danger risk, then you can start managing the population of the Erwinia bacteria in your flowers by putting on these biologicals and preventing the buildup of bacteria in the first place, so that you don't have to battle them after they've already built up. So the model does give you a heads up that there's danger coming, and that's when you want to get started with your integrated program.
I agree. So here, just a couple slides, trials we ran last year, sort of trying to put these ideas together. You can see that most of the treatments in this slide have Blossom Protect as the base, and then after Blossom Protect, we varied it in some way. So it could have been Blossom Protect, then Quava, 3 quarts, or Blossom Protect, then Serenade Optimum at its full rate plus Quava at 2 quarts, or Blossom Protect, then just Serenade Optimum, or just variations of Blossom Protect alone. And then you can see below that is Serenade Optimum twice, just by itself, no Blossom Protect, and as Tim said, it's not quite as effective as a material as Blossom Protect and the coppers have been, and you can sort of see that in this graph, and then antibiotics and then water. And this was kind of a light pressure trial for us, 12 strikes on the water treated control, and you can see that it's not statistically different than the other Blossom Protect treatments, but Blossom Protect once by itself was the very best treatment in the trial. I think we had 1 strike in the 4 trees that were treated with this material. And so that's why you get that like quarter strike per tree or whatever that number is there. Very, very few strikes, but all these other products are all these other treatments here are statistically equivalent. So in this trial, Blossom Protect was the only thing that we needed to control fire blight in this trial, in this pair trial, in this year. We will show you a second at that we have a fairly wet climate in late spring in Corvallis, so when we get into the period after petal fall, which is when the fruit are expanding, which is the period when russet develops on the fruit because the cuticle gets so thin with the rapidly expanding fruit. This is when russet sensitivity goes up, and moisture plays a big role in this, and so and I'll show you the data in a second, but we could attribute a bit of russet in this trial to the Blossom Protect, which we don't see in dry climates, and a bit more russet to Quava, and again in a moist climate, coppers on the fruit are prone to inducing russet. Next slide. So here's another trial. This is the opposite. We ended up inoculating this trial twice. And we didn't need to. We thought we did, but we didn't need to. And we ended up with 400 strikes. This is Gala apples. Now, 400 strikes on the trees, and that's a lot of strikes. And you can see here, streptomycin took that down to about 120. So that was one of the better treatments in the trial. And you can see here, Blossom Protect by itself under this high inoculant pressure it had a response, but it wasn't what we'd like to see. And then you can see that as we look at the curve here, as we add the Serenade then, and we add Serenade and Quava, we see that we're taking this down. and then Blossom Protect plus Quava in a real high risk, high inoculum pressure situation was almost as good as the streptomycin. So those other treatments that we're adding on top of the Blossom Protect later in bloom are having their impact in a higher pressure situation. Next slide. Next slide. Okay, so just talk a little bit about russet. And the first person that reported about russet with Blossom Protect was Stefan Kuntz in Germany. It's the Lake Constance region in Germany. And Stefan is the sort of the inventor of Blossom Protect. And he published this graph a number of years ago, but he just showed that when the number of application gets up, and this is a fairly humid production region in Germany, it's not I think the most humid, but it's certainly compared to western U.S. it's a much more humid area. And he showed that once he got his applications up to this 3 to 4 applications of Blossom Protect, the russet incidence started to go up. And that's a little bit what we're thinking about when we say our recommendations of up to 2 Blossom Protect treatments, is that we don't want to get up into this number where we're just relying on one material all the time. So we feel that if you can mix these materials, put Blossom Protect on as a base, and then switch to fewer applications of, say, a Quava material, that your russet risk will go down because you're not using one material so many times, and that's kind of the basis in our recommendations. The russet with Blossom Protect is actually an infection of the yeast under the cuticle of the fruit, and so it needs a wet period like apple scab type wet period, and then you actually get the yeast growing just like you do with an apple scab infection. So in a dry environment like Yakima or Wenatchee, and Tim will probably say this, he's never seen Blossom Protect russet fruit. They also don't have apple scab, but if you get into a situation where you've got long wetness periods with apple scab, then you might see some russeting with Blossom Protect.
Can I just jump in here?
Yeah. Mm-hmm.
So even on the North Coast, we have multiple microclimates. So we'll tend to see less russeting where there's a drier climate, as indicated by leaf wetness. As Ken said, really leaf moisture, leaf wetness is the real critical issue. But where you get into areas where you have that are scab prone and have higher leaf moisture, even in the same county, you will see more propensity for russeting than where you have where you have less leaf moisture. So it's not just state to state; it's even within individual growing areas in a smaller geographical range.
Yeah. Next slide. So here's a summary. Rachel and I have been trying to sort of like highlight this risk, and we're doing a bunch of these trials again this year. So basically the same program that I had before, starts with water, then antibiotics, then Serenade, and then Blossom Protect or Blossom Protect followed by Serenade. That's the crosshatch bars. And then Blossom Protect and Serenade plus Quava. So the dark bars all have Quava in them. Then I put a Luna Sensation treatment in there, which is a fungicide. It's not organic, but we were trying to keep the yeast from spreading to our trees. So you see that the Luna Sensation is a pretty good looking treatment in a couple of these. But so this is the level of russet that we get. We go out and measure the fruit, and there's some examples of russeting on fruit down in the lower right there. But you can see the water and the strep and the Oxytet and the Serenade, the clear bars there are generally the safer treatments. And then in Corvallis on barlett pear, you can see we bumped it up a little bit with some Blossom Protect, and that we have a wet environment again. And then with the black bar in a wet environment, that's Quava, and we're bumping it up more with coppers in this wet environment. The next box is Camise pear, and Camise pear, you just sort of blink at it or wink at it, and it wants to russet. It's kind of the poster child of fruit sensitivity to russet. And you can see here the same treatments going across, water, strep, Oxytet, Serenade, the lowest levels of russet, and then you get into the Blossom Protect, and not really much of a change, and this is a dry environment, and so there's not a lot of water around, so you don't really see much of a change there. And then you see the Quava on this very sensitive cultivar and the the picture to the left at the bottom shows some queva, some russeting on camise with queva. So in a very sensitive cultivar, we're getting some russet out of these copper materials. Luminescence there is the best treatment, and so that may represent the closest thing we have to a control. And there is, of course, as you showed in the earlier slide, the Blossom Protect yeast is spreading around on all of these treatments. So even though I say these clear bars didn't receive Blossom Protect, they do have a bit of yeast on it. And here we try to prevent it with a fungicide. So maybe we are getting a little bit out of Blossom Protect on this cultivar, but it's not very much. And then we move to Bartlett, which is fairly russet-tolerant cultivar, and in Lakeport, which is very dry. And you can see that under these really dry conditions, Rachel's producing some pretty nice-looking pears, probably why there's a pear industry in Lakeport, and less than 2% from all the treatments. So the moisture factor is really important in russet, and then the second thing is the cultivar factor and whether the cultivar is sensitive to russet or not. Next slide. So we've come up with some cultivar-specific recommendations, and most of these pertain to pears. So when we're talking about, say, Anjou pears or camise pears, that we're just telling people to be a bit leery unless they're in a really dry environment of materials that can russet their fruit, and Blossom Protect and queva have shown a bit of that on really sensitive pear cultivars. And so the alternative would be Bloom Time Biological and Serenade, though not as effective as Blossom Protect and queva, of course. You're giving up efficacy for safety in this case. And so in a really dry environment, like as we just showed in the last slide, though, that Blossom Protect and queva should be fine on most pear cultivars, maybe not camise, but most pear cultivars. So in more russet-tolerant smooth-skinned pears and apples in a wet climate, we're a bit nervous if you're packing for fresh market with coppers. The risk might be unacceptably high, and that Blossom Protect should be restricted to this early mid-bloom period just to be safe. This is in a wet climate. And in a dry climate, like I said before, we're trying to create programs to minimize Blossom Protect and queva applications. So 1 to 2 applications of each is better than 3 to 4 applications alone. And the last thing and this kind of came out of a problem that we had last year, was that we had a boss grower in California that put copper on to actually enhance russet in a boss, which is what you want. But then they put the Blossom Protect on top of the copper, and we think that the acidic buffer from the Blossom Protect went onto this copper and freed a lot of copper ions, and then sort of created a super russeting situation. And so we're telling people to sequence their copper to follow the Blossom Protect and not the other way around, because if they put copper on the trees first, then Blossom Protect with the acidic buffer, they could damage and their fruit. And we don't think that this happens the other way around, because the acidic buffer doesn't really maintain that acidic condition on the plant surface for very long, maybe a day or two or so. Blossom Protect first and then copper, and not the other way around. Next slide. So we're back to this, the recommendation slide, and this is basically the last slide, and I'm going to get Tim and Rachel back in here and see if they have anything to comment here.
Let's see. Let's see, what should we say? So pre-bloom, fixed copper, sanitation. If fire blight was in the orchard last year, I think we've seen a tremendous increase in the number of those applications. Personally, I feel that if there is a larger growing area of organic pears that people have adjacent orchards, that this is an excellent area-wide type of a program. So that if you have a neighbor that's really dirty, even if you're pretty clean, if you could get everybody and all your neighbors together to put this copper application on in a sort of an area-wide manner, I think would be a pretty good strategy overall.
Yeah, I agree with that. I think our curves that we got in those studies would have been even better because we were just doing a 4-acre block inside of an orchard, and by the time you get to late late petal fall and things like that, the bees have gone all over the place, and if that had been area-wide, I think we would have seen an even greater response.
Yeah, these tests have shown that this yeast gets around.
Yeah.
And the untreated checks and that blossom infestation study that we did, the untreated checks had 50% of the blossoms with the yeast in.
Absolutely.
So this is the only product I can think of that would be effective if not perfectly applied. With most products, we like to see every blossom get treated, and it doesn't take quite as perfect a treatment for the organism to make it into the flower. Bees will carry it in,
yeah,
or other pollinizers.
Yeah.
So that would be an area-wide potential for both the pre-bloom fixed copper sanitation treatment and also the blossom protect. So an area-wide type of a strategy is good for those of you who are already doing area-wide programs for insect control.
Well, one thing, if your neighbor uses Blossom Protect, you're going to get it too.
Exactly. Yeah, that's true. Absolutely.
Next slide. I do think timing is important, though. You want to get it in as well as you can in your own flowers, just waiting for your neighbors.
Right.
Unmuted.
Doing your own spraying.
Yeah.
Yeah, it wouldn't be adequate unless you sprayed your own orchard.
So I got to say, we're going to open this up for questions, and you can see the whole list of questions there that you can be thinking about, but I just have to say something about our sponsors. And so eOrganic has done this now for us for 3 times, and it's really awesome in the extension. And funding for that comes from NIFA, USDA NIFA, which is the National Institute for Food and Agriculture, of which we are all on a project together. And so NIFA has really facilitated this research very well. And then we have our grower group partners, which is USA Pear, and the Fresh Pear Committee has helped with this project, and the Washington Tree Fruit Research Commission has funded us consistently on this, and so has the California Pear Advisory Board. And so those are grower groups that have funded us. And then you can see our universities there, of course, Pear Salary, and so they're supporting us as well. So we're going to open this up for questions, Alice, and see what's on people's minds. So.
All right. We're about to begin question and answers. So for anybody who missed the very beginning of this presentation, you can just use the question box on your GoToWebinar control panel and type in questions and hit return. And if the question box is closed, you can just open it by clicking the small plus sign or triangle next to the word question to open it up, and we'll answer as many as we can. I also want to mention that we really value your feedback, so we would very much appreciate it if you could fill out the follow-up survey that you'll be receiving in an email later today. Also, this webinar is being recorded, so if you want to share it with others or see something again, you'll be able to find it on our website at extension.org. I'll pull up the link in just a moment, and we have the archived webinars from this group's other presentations there as well. Yeah, let me just pull that up here. And if you have additional organic farming questions that are not necessarily about fire blight, you can use the eExtension Ask an Expert service, and you'll get an answer. So we do have a bunch of questions coming in. Let's see. This is just a consumer question here. It's not directly related to fire blight spraying, but this person wants to know if the organic apples that he has are safe to eat and have been treated with antibiotics, or if there still has not been a change. He loves apples and firmly believes in organic, non-GMO food, so he just wanted to know when they're antibiotic free.
Well, first of all, if they're fruit that you bought this winter, they were produced in 2014, so antibiotics were still legal to use in organic farming in 2014. So there was a potential that the trees were treated with antibiotics last spring. That said, not nearly. I think, I don't know, Tim, did you do this? I mean, it probably maybe a third of all tree fruits ever get sprayed with an antibiotic in any given season. And lastly, antibiotics have a very, very short residual. We don't even think they're there after 3 days after treatment. So that they're gone, they've degraded away, and so the materials don't have a very long residual at all. And but so we're talking about spraying flowers in April, and then we talk about picking the fruit in September or October. There's just really no measurable residue of antibiotics.
I've never seen any data where any residue was ever picked up in any fruit.
Yeah.
Even when they sprayed it late on purpose to test the residues.
So that's the safety as we perceive it. Organics, the NOP has said no more antibiotics in organic production, and so coming to 2015, you can be guaranteed that antibiotics have never been sprayed on the tree from which the apple came from.
Now, if an organic grower sprays antibiotics, that limits his market. There's many markets that won't accept antibiotics sprayed, so by far the largest percentage are not sprayed at all.
Right. Yeah, that's right. And in 2015, if they are NOP certified, they will not be treated with antibiotics. It's illegal to use antibiotics in NOP certified.
Okay, great.
Yeah. Say that is, if they are NOP compliant or certified, so that if they're not, and it says natural or something like that, there's no regulations or anything. So you have, the growers are very different about how they describe their product.
Exactly. Yeah, and in a conventional orchard, you can still use antibiotics.
Right.
But I would say that since we've started this work in organics, that there's a lot of conventional growers that are now using Blossom Protect and using some of the approaches that we're talking about in the webinar here. So it's a cross-current thing. I think overall the use of antibiotics will likely go down as other alternatives come on, and I think we're all seeing that. So.
Right. Russeting is really the biggest issue for most growers.
Absolutely. Yes.
Okay, thanks. I hope that clears that question up. Can we imagine a strategy with Blossom Protect and then kill off the yeast with lime sulfur so as to avoid russeting?
Mel, I'm very interested in that question, and that's a kind of a research topic of ours. A little bit nervous about lime sulfur late in bloom, and you see like a lot of scab programs in the Midwest will use lime sulfur in early bloom, but then as they get into later bloom, they switch to sulfur, and I think part of the reason is that lime sulfur itself can be a bit of a russeting agent, and I think that maybe if you're from the Midwest and you're asking this question, that you might check with somebody that's knowledgeable about organic scab control, and I guess that's not really my forte, but I think it's a really interesting question, and there's some other materials that I think we're going to look at this year for that reason, and one is these dihydrogen oxide materials like oxidate, which are oxidizing, sanitizing agents that we think are very antimicrobial, and they do have some effect on fire blight control, and we're also going to look at them as what effect that they have on yeast populations. So that's definitely coming in our trials for this year.
This is Rachel. Any use of sulfur product during the time that late when fruit becomes susceptible to russet is going to give you a risk of russeting your fruit. So that is going to be the case, and again, the same criteria apply. If it's a very dry situation and you don't have moisture, then you're going to have less of a risk of russet, but organic growers that are using on a sulfur program, that is the risk they take, is it's pretty hot, and they can get a russeting problem with sulfur use, and lime sulfur for sure.
Okay, so what would be a typical program using lime sulfur and Blossom Protect?
You're talking about using it then as an apple grower for thinning, is it like Ken said?
I don't know. The person said. Okay,
yeah.
thanks.
The typical program is it's in apples, not in pears. And the typical program is when the king blooms are fully open is when the first treatment goes on, the assumption being that you've set your king flowers. At that point, they're pollinated. It happens very quickly, the pollination. And then and so you burn off the flowers in the secondary in the cluster. The king flower is the desired fruit because it's the strongest flower and it's going to give you the biggest fruit. So that one's open. Then you burn off the other flowers around it as they come open. So the first treatment occurs after the king bloom is open, and then again 3 days later. So that's about 70% bloom. So 20 and 70% bloom is the lime sulfur timings to try to thin fruit, and then after that is when your fire blight risk then can build, and so for the flowers that are left, and of course you don't burn out every flower, the flowers that are left, so the blossom protect would go on even a day after the lime sulfur. I sometimes tell people if it's a big orchard, well, you could probably just start the sprayer, at the other end with blossom protect, because lime sulfur has a very, very short residual, and it isn't going to last very long out there, and just hours to do a day maybe. And so the blossom protect goes on immediately after that second lime sulfur treatment, and then you put in your blossom protects in there growing in the flowers to protect them.
Okay. Do you have any concerns about bee health from copper sprays?
Tim, you want to answer that one?
No. I don't think that the copper sprays are hurting the bees.
I don't either, and I guess I've never seen any data on that. The coppers that we're speaking about here and using them during bloom have such a low amount of copper in them. Quave, I think, is 1.8% copper, something like that. It's a very, very small amount of copper. I don't see a concern there.
Okay. Okay, here's a question, I think from a non-organic grower. Do the antibiotics like Oxytet used for fire blight control kill or prevent biologicals like Bacillus subtilis from controlling fire blight, or can the antibiotics and biologicals be used in conjunction in non-organic orchards and be tank mixed?
Yeah, well, I don't know if I want to answer this question because this is an eOrganic.
It's an organic webinar.
Yeah. I think we'll if they want to, they can write me an email, and on your handout, my email is there, and I'll happy to answer it then.
Okay. Let's see. Do you know of any trials going on in the eastern USA?
Yeah, George Sundin at Michigan State University also has funding from the National Institute of Food and Agriculture to work on non-antibiotic approaches in Michigan and, by extension, the states around Michigan.
And Keith Yoder.
Keith Yoder does some work in Virginia as well, yeah. Mm-hmm.
Okay, thanks. In areas with long bloom periods, such as California, which has about 4 or 5 weeks of bloom, and in orchards with many varieties in close proximity, how should the program Dr. Johnson recommends be averaged out? In other words, should all materials be used at the beginning of the first bloom or later on?
This is a very hard question. This is the hardest question, and the person that asked that I bet knows that this is the hardest question. So I've run into this one a few times now, mostly from people from California, and it's a very difficult question. I guess because the lime sulfur, they describe our bloom in Oregon and Washington as popcorn bloom. It happens pretty fast and pretty quick, whereas bloom down in areas farther south, lower latitudes, can extend for quite a period of time. And so it does just get more difficult. And I do think that biologicals are very important in that kind of a situation, and get your blossom protect out there and growing and spreading flower to flower, and it's going to keep working for you during that whole period. If you're going to run out there then with lime sulfur and kill off your yeast and also probably your pathogen, then you probably need to think about reapplying that blossom protect, and that's just the fact that the lime sulfur is toxic to microbes as well as to burning out flowers. And then the frequency of other materials, the programs, I know pear growers in California that will spray 8 to 10 times with a material like an antibiotic or a serenade just to keep that pathogen in check in those epiphytic curves, and so it means a lot of spraying, and I can't say I have the exact answer, and everybody's situation is a little bit different, but to me having active biological out there is going to be useful.
And in principle,
think you're definitely talking about a 2-spray program. If you've got something like Bartlett and Bosc, you're going to time your first treatment for 70% on your earlier variety, and then you're going to reapply when you get to enough open blossoms on your later variety. And then in between, you're going to have the material that you sprayed for the earlier variety start to cover the blossoms of the next one and the later blossoms of the first one. But you're probably looking at 2 sprays.
Biologicals tend to spread best when there's lots of flowers out there. So as you drop off on the flower count, you may have to make up for that by spraying rather than relying on the pollinizers moving it around.
Yeah.
Right. I would think that for most orchards here in California, if you're definitely looking at the earlier bloom versus the later bloom ones, you're probably looking at 2 treatments.
Okay, when you're talking about copper interacting with Blossom Protect, my understanding is you're talking about bloom applications, not coppers applied at delayed dormancy. Is that an accurate understanding?
That's right. Yes, correct.
Okay. Let's see. Is it the natural product chemistry or the living bacillus that enable products such as Serenade to provide control of fire blight? Any idea how long the bacillus persists in the flower?
Yeah, the products that are based on bacillus, we don't think that the living bacteria that you buy when you buy the product has any role in control at all, and that most of the living product these are gram-positive bacteria, and most of the living agents in the bag are long-term resting spores of gram-positive bacteria, and they're very slow to germinate, and they rarely grow. You can get at them to grow in a petri dish, but they're very slow to germinate, and we don't really think that the living bacteria in a product like Serenade or Double Nickel has anything to do with control. And it is all the natural product chemistry in the flower that does the work. So when these things are grown in fermenters, they are making these antimicrobial molecules or small proteins for the most part. And when they dry these products down, they're very careful to preserve those antimicrobial peptides or proteins. And so when you spray the product, you essentially have something that's very similar in how it's made to an antibiotic. Antibiotics are made the same way, and that these antimicrobial peptides are the thing that do the work. What's different about the bacillus products from antibiotics is that the antimicrobial peptides are quite short-lived. So we think you get about 2 days of efficacy out of a Serenade, and a lot of times 2 days is enough. But sometimes it isn't. So the frequency of application, you've got to be thinking about putting on, say, Serenade every 2 days in a high-risk situation, whereas with an antibiotic, it might have been every 3 or 4 days where you get an effective residual out of those materials.
Okay, we have somebody who commented as an answer to a previous question. Carrick Cox at Cornell has also been testing Blossom Protect for several years, so thanks for that.
Hi, Carrick.
Okay, it wasn't him?
Oh, it wasn't him.
No, that's okay. Anyway, let's see. Does Cueva or Previsto have an effect on scab control?
Rachel, do you know the answer to that? I think yes. In fact, I get scab in my trials here, in my apple trials in particular, and yes, it does. I don't know relative to, say, to sulfur or whatever, if it's better than sulfur or not, but yes, it does. It's a general fungicide, Cueva, and bactericide, and so yes, you'll get some suppression of scab with it.
I would agree. I don't think we really know. I mean, copper is a material that is used. It is a general fungicidal, bactericidal material. What how much, it's more probably going to be a suppressive, but there is data for apple scab. I know my colleague Paul Vossen in Sonoma County did some work years ago, handgun work with copper on apple scab. I have not personally done trial work with copper with pear scab, but probably worth doing. And yes, I agree with Ken. probably the higher metallic equivalent that's out there, the more effectiveness efficacy you're going to get. Those materials are fairly low metallic equivalent, so you're probably not going to do as good as you do with something with a higher percentage of fixed copper, but we don't really have that data. If you go back into the real old literature prior to these newer fungicides, you're probably going to find some of that information in the real old literature on using copper for scab.
I just added that Serenade is also fungicidal and would probably give some protection to scab. I can't say how much, but I know that Serenade's used in banana production and things like that for control of black sigatoga and things like that. So there is some value to Serenade for apple scab control too.
Ken, I have a question. Could you comment on Previsto, since it's come up a lot about the registration situation?
Well, yeah, it's still held up with EPA, though OMRI has already approved it as an organic material, but the EPA registration has been held up until they do one test on one of the inerts this summer, and they're expecting to have that done and completed sometime in late 2015. So Previsto, it should be registered in 2016. So we're a year off from that material.
Right. So when you go talk to your dealers or your PCA, you're really on the solubles. You're really talking about Cueva.
Cueva right now, yeah, and then next year Previsto, and Previsto's look really good in fruit finish trials in terms of its fruit safety, better than other coppers, and so that should be a useful material for people.
Okay. Let's see. When you talk about the letter A for 5 to 6 pounds per A, did you mean acres?
Yeah, acres, right. And generally, when we talk about these trials in fire blight, most of us are working under the assumption of 100 gallons of water per acre. And I know there's some baby trees that's too much water, and I know there's some great big trees that's too little water. So generally, when we're talking about per acre application rates, is the big A means per acre, and it also is assuming that we're mixing at 100 gallons per acre.
Okay.
So you would look at the rate per acre that we give, and use the rate per 100, and cover the trees so that the blossoms are well covered.
Yeah, most of our trials are handgun trials, so we're not even, a little tiny individual tree limb kind of thing. So we're mixing at this rate and trying to get it to this state we call near runoff, which is about what an air blast sprayer can do, applying that rate per acre. Yeah.
Okay. Is Blossom Protect labeled for wine grapes, and if so, what is the mortality or fate of Blossom Protect post-application? It's sort of an enology question. Here.
We can answer that one. It is labeled, but it's labeled as under a different label name, and it's labeled Botector. And so let's look that up. Westbridge website would have some information about that. Basically, it's the same yeast that's in Blossom Protect, but you don't apply the buffer. In terms of how well it works, I don't really know. I've never worked with it, and what kind of numbers of yeast you get in a grape cluster, I don't know the answer to that either. But Westbridge, if you contact somebody from Westbridge, they might have an answer to that.
We should also point out that the Aurea basidium pullulans is a natural organism that occurs more often on flowers than any other organism that Dr. Larry Pusey was able to find when he was surveying organisms on flowers. It's very common out in nature.
It is very common, and there's some strains. We've run into a couple of problems where people have seen problems with Aurea basidium pullulans causing problems on fruit, and one was russeting of apples, and another was a post-harvest storage rot of cherries. And we've got the ability now to be able to molecularly determine the Blossom Protect strains anyway, and so we've looked at those situations and wondered if it was Blossom Protect spilling over into other crops or whatever where these situations occurred. And to the best of our knowledge, these were not situations that were caused by the Aurea basidium pullulans and the Blossom Protect but just by the wild strains that are out there. It's a very, very common organism.
Okay, I think we have time for one last question here. Let's see, how fast can fire blight spread through an orchard?
That's your
Like
Tim.
It can go like fire. And sanitation is the primary 90% of the situation when it comes to controlling fire blight. If you have cankers in your orchard, everything is going to be bad from that point on. You don't need as much heat. All you need is a little water and flowers, and you're going to have a fire blight problem. And these products that we've been talking about are good fire blight control materials, but even a perfect fire blight control material won't stop fire blight if you have a lot of cankers in your orchard. So the winter when you can see the blight cankers the best is your primary fire blight control period. Everything's just going to go downhill fast, and we've been able to document one canker affecting an entire acre of orchard.
Yeah, fire blight is a sporadic disease, so it sometimes it can just go like lightning, and other times you'll think you wasted your money, and you probably did, but it's often very hard to tell the difference about what kind of condition you're going to get. So it is a sporadic disease, but it's a very destructive disease, and over the years that we've been involved in fire blight, I think we've tried to steer people a little bit more toward thinking about programs, and like Blossom Protect being a big component of that in terms of doing a little something like every year for fire blight, and I think it pays off.
There is nothing that works on fire blight after the fact of infection.
That's exactly
Despite
right.
the fact that we continue to have lots of people selling products for after infection treatments. Yeah. Nothing works.
Yeah.
Okay, well, we're running out of time, so I'd like to thank everyone who asked questions and say once again that you can find this and our many other upcoming and archived webinars on organic farming and research topics at the links on your screen. So thank you so much, Ken, Rachel, and Tim, for joining us again today and presenting your work, and we hope you come back again with future updates.